There you go.
Yeah? Fantastic. So firstly, thank you for that lovely introduction, and actually thank you so much for inviting us here today, and even more for suggesting that Group B Strep Support is one of the is the charity that you're supporting this year, for all the cakes. I'm so excited about the cakes, and for all you are doing and have done to raise money. You know, huge thanks to Melanie, to Kirsty, to everybody. I've been given 5 minutes, so I will crack on. My husband and I started the charity Group B Strep Support 26 years ago after our middle child died as a result of Group B strep infection. This is one of my most precious photos. So you've got there on your left, my firstborn, Oliver, who is here with us today and has given me permission to share this photo. It's not a surprise to him. And then you've got me me holding Theo and my husband, Oliver. So my husband, Robert. So let's do that one again. Oliver, me, Theo and Robert. And Theo died 17 hours after he was born. And we learned very soon after that most of these infections in babies are preventable. And so we were fired up to just try and stop it happening to somebody else. That was really all we wanted to do. So we decided the best way to do that was to set up a charity. And I confess we didn't think it was going to take 26 years to get to the point we are now, but this is where we are. So the charity really does 4 things. The objective was then, is now unchanging, to stop this happening to babies where we can. So we provide information and support to families. We provide information and education to health professionals. We campaign for improvements to particularly UK policies and practices, and we support Group B strep research. And we can do all this only because the work that we do is underpinned by an expert medical advisory panel. And I'm sure many of you will know some of these people. So we have on the medical advisory panel experts in the key areas around Group B strep. So obstetrics and gynecology, neonatology, microbiology, vaccinology and epidemiology, and last but by no means least, midwifery. So in terms of the information we provide, as I hope you will have found, we have a website. It was visited last year by just short of 500,000 people, and that has a whole host of information on there, not only aimed at families, but also aimed at health professionals. We provide leaflets for new and expectant parents and also for health professionals. And of course, we provide training to health professionals particularly. We have a helpline which provides information and support, not advice. We absolutely don't give advice, but we provide information and support through whatever channel somebody tries to reach us on. So social media, phone. In the old days, we used to get letters. We don't get so many of those now, but we do get emails and all kinds of social media. And that helpline provides high quality information there and then. We also have some peer support groups, so families who've been affected can talk to each other and share with each other. We offer a listening ear, and of course, we are not experts in all areas, so we are very happy to signpost to other organisations where we don't have that particular expertise. So organisations like SANS or Miscarriage Association or whoever. We campaign to improve UK policies and practices, and our current campaign is to support the GBS 3 trial, which is ongoing at the moment, which is looking to compare screening, 2 different screening policies with the UK's risk-based policy. So I can't resist asking whether St George's is taking part, and if it isn't, please would it. But really, in terms of our campaigning work, anywhere where there's any sort of consultation relating to policy around Group B strep, we're there, including, as Kirsty alluded to, the WHO. We were delighted to have be involved and to influence that in some way. And patient and public involvement. I mean, that's really how we get involved with research at the moment. We don't have sufficient funds so far. never say never, but so far to fund research. So most of our research involvement is through PPI work, and those are some studies that we're involved in, and I'm delighted and privileged to be working with Paul and Kirsty on a number of projects. We have done some surveys to find out the impact of Group B strep infection on families, both late onset and early onset, and we published those results some time ago. And of course, we now run annual conferences. Our next one is coming up next week. I am delighted to say that both Kirsty and Paul are involved in that. There's still time to get tickets. It's a 2-day conference, good rates, and that was really all I wanted to say. Thank you again. That was a whistle-stop tour of what Group B Strep Support is about and what we do. Oliver, Jane and I will be here for the duration, so do come and speak to us if there are any questions or anything you'd like to know about the work we do. And thank you once again.
So I'm going to hand over to the first of our speakers now, Professor Paul Heath.
Thank you, Kirsty. Rakan, are you on standby? Is this me? Yeah. So, yeah, good. So, yeah, thank you very much. It's obviously a pleasure to be speaking with you, and again, very much thank you to Jane and to the Group B Strep Support Charity. We've had the pleasure of working with you over a number of years now, and it has been and continues to be a pleasure to do that. So, in fact, I was asked to talk about vaccines, and in some ways talking about vaccines now is a bit premature because some of the other talks actually set the scene before we get to the vaccine bit, which of course we all think is the ultimate solution to this problem. But what is the problem? And I've only got one slide to demonstrate the burden of Group B strep, and this is the global burden of Group B strep, and this comes from a nice review involving multiple papers and systematic review and meta-analysis that Kirsty and I and others were also involved with, that was led by the London School, trying to define the global burden of Group B strep. And so you can see some of the numbers here, which are, of course, sadly impressive numbers. So if we start on this side, we have 21 million pregnant women colonized with Group B strep. And just to remind you, Group B strep is commonly carried in the gastrointestinal and vaginal tract of otherwise healthy pregnant women. And it's from there that it's transmitted to the infant at or before birth to result in early onset disease, which is disease in the first week of life. The other burden of disease is the period between the first week of life and 3 months, and we call that late onset disease. And I know Costas is speaking to this later on. And that is also clearly an important burden of disease, particularly because most cases of meningitis occur as part of late onset disease, and meningitis carries with it a huge and important morbidity and mortality. So maternal disease occurs as a result of Group B strep, and you can see the estimates there. At least 33,000 maternal invasive Group B strep disease cases, 57,000 stillbirths per year, 90,000 infant deaths, mostly in the neonatal period, the first 28 days of life, and at least 320,000 infants with Group B strep sepsis and meningitis, and then all the developmental disability that occurs as a consequence of both meningitis and also, we now know, simply from Group B strep sepsis itself. So there is a huge and global burden of Group B strep, and we all believe that the solution to this is through vaccines. So this is a cartoon of the organism, this encapsulated bacterium. So you can see in this cartoon some critical and important structures that are important for the way this disease occurs, but also in terms of as targets for vaccines. So particularly the capsule, which is capsular polysaccharide, a sugar capsule, of which there are 10 different types, 10 different serotypes. And that becomes important when we think about vaccines based on the capsule, but also a number of important surface proteins there in the cell wall that are again important in the virulence, the way this bacteria causes disease, but also in terms of immunity. And these surface proteins are also a target for vaccines, as I'll show you. But the rationale for developing a vaccine goes back to the 1930s, where Rebecca Lansfield demonstrated that protection against infection in mice in one animal could be achieved by antibody against the capsule of the bacteria generated in the different animals. So capsular polysaccharide, CPS specific sera. And then in humans in 1976, Carol Baker showed that those babies who went on to develop GBS disease were much more likely to have low levels of antibody against the capsule of the bacterium than those babies who didn't go on to develop Group B strep disease. So raising the possibility, of course, that by increasing these levels of antibody, disease could be prevented. Now, I mentioned the capsular polysaccharide approach. Important, therefore, to recognize that there is, in fact, 10 different types of capsular polysaccharide. And this becomes important when thinking about capsular polysaccharide-based vaccines. So there are 10 different serotypes, and this shows in this pie chart the different proportion according to the different types. But perhaps you can see that actually overall around 6 of the serotypes account for around 95% of invasive disease. So a vaccine based on 6 serotypes would cover nearly all of the cases. And in fact, this approach based on the capsule has been the approach to develop a vaccine for many decades. And in the 1970s and 1980s, there were a number of different capsular polysaccharide based vaccines that were developed in the US, mainly led, in fact, by Carol Baker and her team. And these vaccines were initially just the capsule, and then it became clear that the capsule alone wasn't sufficiently immunogenic. So the capsule was then joined to a protein to develop capsular polysaccharide protein conjugate vaccines. And the proteins originally chosen were tetanus toxoid and subsequently other proteins. And as you can see, there was a lot of development over these years, including using these vaccines in pregnant women. And this is the first pregnancy study with a serotype specific polysaccharide protein conjugate vaccine. And the serotype here is serotype 3, which is the most common serotype, and the protein was tetanus toxoid. And again, this is the Carol Baker group. And the point of this is simply to show that this is this is pregnant women. They were vaccinated at 30 to 32 weeks gestation. They had a very good antibody response. And importantly, this antibody crossed through the umbilical cord, then into the infant out to at least 2 months of age. There's a control group here. You can't see their levels of antibody, so very immunogenic. And this does, of course, raise the question as to how much antibody is needed in the infant. And there are, and we'll talk about this in a moment, there are, this is subject to ongoing work, but there are various estimates of the amount of antibody. This is one estimate, but you can see that actually the antibody levels required to achieve protection against early onset disease are much lower. So these antibody levels would certainly be adequate to protect these young infants out to at least 2 months of age. Subsequent studies with a 3-valent, remember, ideally we'll have a 6-valent vaccine. This is with a 3-valent vaccine, just showing that it's immunogenic in pregnant women. And then the antibody levels are out for the 3 different serotypes out to 90 days of age, showing well above that of the placebo group. So again, providing confidence that such an approach would both prevent early onset disease in the first week of life and late onset disease out to 3 months of age. So this is the current pipeline. So the conjugate vaccine approach, which is now headed by or led by Pfizer, with their 6-valent polysaccharide protein conjugate vaccine, and that's now in pregnant women. Others are behind, except for Minovax, and they have a protein-based vaccine, and that too is now in pregnant women. And really importantly, as Kirsty said, and Jane also mentioned, the WHO have adopted Group B Strep and have developed guidelines for implementation of vaccines and a roadmap for subsequently the global distribution of such vaccines. So this summarizes the Pfizer approach. So the vaccine is now in pregnant women in a trial in South Africa, but also including our site in the UK. And they're also starting a study where they're giving it at the same time as the pertussis-containing vaccine, first of all in non-pregnant women, but then ultimately in pregnant women. But also importantly, we at St George's, in a consortium called the Prepare Consortium, which is led by Kirsty, are also doing a trial with this vaccine in pregnant women in Uganda, and this is about to start. And this is particularly addressing the issue of HIV infection and how that influences the immunogenicity and safety of the vaccine. The Minovax, the protein-based approach, these are the proteins that are the focus of this vaccine, the alpha-like proteins, of which there are 6. This, in fact, is focused on 4 of them, which covers nearly all pathogenic strains of Group B Strep. So the 6-valent polysaccharide protein conjugate covers around 95% of cases. It's thought that these 4 alpha-like proteins will cover all of the strains of Group B Strep. This is their program. So we too are in fact, this study, again, through the Prepare Consortium, has just finished recruiting in South Africa and Uganda in pregnant women, again focusing on HIV positive and HIV negative women. This is another study, and again we're involved in this study here at St George's, which is in Europe, and then there's another study. So all of these are in pregnant women. So you can see that these are progressing. They're now being given to the population that needs these vaccines. So the next question is actually how to license such a vaccine, and this is the next and most important barrier before implementation can occur, because such vaccines need to be licensed by the various regulatory bodies around the world. And the classical approach is an efficacy trial, but actually that needs a lot of pregnant women and is going to be a very big and very expensive trial. Not impossible, but a challenge. The preferred approach is based on what we would call serological correlates of protection. So identifying the amount of antibody that is required to protect infants against Group B Strep, and then simply ensuring that these vaccines can achieve that amount of antibody and do so consistently and in a range of populations. And this is the approach, in fact, taken in the UK with the meningococcal C and meningococcal B vaccines. They were licensed based on serological correlates of protection. So we think that this approach will be the most appropriate approach for licensing Group B Strep vaccines as well. This just shows, these are South African data, showing that we can identify levels of antibody that are associated with protection. This is for 2 different serotypes. And the same can be said for the protein-based vaccine, actually. These are data showing that there are levels of antibody that seem to predict, natural antibody that seem to predict protection. So this is the most attractive pathway for licensing vaccines. There's general agreement from the key regulatory authorities. We need to really pin down what these levels are, and we and others are involved in large correlative protection studies. Or the one that we're involved with which Costas, who's on the line, is coordinating, is called iGBS3, and it's run on the back of the GBS3 trial that Jane mentioned earlier. So to bring us back to the potential of such a vaccine, this again from this modeling work shows that the current approach, which is giving antibiotics in labour, is limited. And as you can see, the potential is much smaller than the potential of an effective vaccine. in terms of the numbers numbers of cases of maternal infection, of infant infection, of stillbirths, of infant and maternal deaths that could be prevented by a successful vaccine. So there is a significant global burden, as I've shown you. The currently available strategies are short-term and indeed not realistic when we start to think about their application in other countries, particularly low and middle income countries. And as with the other significant causes of meningitis and sepsis in young infants, the approach has to be through vaccination, but this time through maternal vaccination. So I would finish by saying we're kind of nearly there. There's a bit of work that needs to be done, which we're very pleased to be involved in doing. But, you know, when Jane and her team return in 5 years, it will be to demonstrate the success of such a program. So I'll finish there. Thank you.
Paul, are you setting up the next person?
No, I can do that. Who is the next person?
Kay.
Okay. Okay,
so,
perfectly capable of doing it.
Yes,
All right.
exactly. There you go.
Okay.
So over to you, Kay. So thank you, Paul, for that. Oh, questions now. Yes, sorry, Paul, you've got questions. Sorry, Kay. Sorry.
Yes. Well, I've got one.
How dare you ask a question?
It's probably too late to suggest this now, but why isn't just giving a bolus of antibody a reasonable alternative to vaccination?
A bolus of antibody. Do you mean? Well, the question would be when would you do that?
Right.
So would that be to all women in labour, for example, or would it be?
It would be. I imagine it would be in infected women.
Yeah.
Pre-labour. I mean, you're giving antibiotics in labour.
Yep.
So a bolus of antibody, I think, would have to happen a little earlier, but then your vaccination has to happen a lot earlier.
Yeah. Well, the vaccination probably should be at least 2 weeks before the baby is born to allow adequate placental transfer of antibody. I mean, an approach in which you were to give specific antibody at some point before delivery could work in principle. It would need to last a certain amount of time, obviously, and that time is just until the infant is born and with adequate placental transfer. As always, there needs to be thought about who the appropriate group is. Is that all pregnant women, or is it selected based on other factors, known colonization and so on? So, I mean, in principle, that's what a vaccine is doing, of course, and it needs to do so for a limited period of time, perhaps unlike other vaccines which need to last for longer. So in principle, Julian, it is something that could work. The fact is that there isn't any such preparation, nor is anybody working on it, to my knowledge. It would need to cover all of those serotypes, of course. So that's a challenge because it's a multivalent vaccine, so it would need to be a multivalent monoclonal antibody cocktail, which again, I guess is in theory possible. But I think they're the factors, and then it would need to have an equivalent cost, I suppose. It would be the sort of health economic issues associated. But yeah.
Thanks.
Any other questions for Paul? Sorry, Paul. Can't see any online. Jane.
Jane.
Fabulous presentation as ever, so thank you. And I was just thinking about the vaccine, and absolutely you know how excited I am about the vaccine being available. Obviously we wanted to stop early onset and late onset, prevent early onset and late onset, and potentially prevent preterm labours caused by Gbs Strep stillbirths, maternal infection.
Yes.
Is there any, I mean, how long is it likely to last? Because if we vaccinate a pregnant woman, and I'm thinking particularly of low and middle income countries where that programme may be more difficult to implement than, for example, the UK or the States.
Yeah.
Is there any chance that you might get a 5-year immunisation?
Yeah, I mean the duration of protection will be one of the questions that will need to be addressed once we have such a vaccine. And of course, if you look at what we do at the moment, so tetanus vaccine in the LMIC setting, of course, is given every pregnancy. In our setting, the pertussis vaccine is given with every pregnancy. So we don't anticipate that a pertussis vaccine will provide protection for subsequent pregnancies. I mean, in time it may become clear that it is, but the starting approach is to repeat at each pregnancy. So I suspect with a Group B strep vaccine, that's how we would start. But by then, we would anticipate we would know how much antibody is required, so we'll be able to titrate that, and it may become clear that there's persistence of antibody. I think the conjugate vaccine studies that have been done till now show persistence of antibody through 2 years after vaccination, so that's a good period of time. But as always, until we know what level of antibody is required, we don't know where that is relative to it. The other thing with Group B strep is that because Group B strep itself is so common, it's entirely possible that one could be vaccinated and then be boosted by being colonized with Group B strep. And that may keep the levels up as well and therefore provide long-lasting protection. So I think it's one of the really important questions that we need to address once we have a vaccine. We could talk more about this because, of course, the other thing that a vaccine might do is actually reduce colonization. Reduce disease is what we expect, but will it have an impact on colonization itself? And we don't know the answer to that. So all of these things, these are questions to be addressed once we have a vaccine, and then fine tune a vaccine program, begin by doing it every pregnancy, which I think would be the way. But then as time goes on, as we learn more, it may be that becomes unnecessary.
Any other questions for Paul? I'm just having a quick look, see if there's any online.
Sure,
no hands up.
Oh, is that?
It's all the.
I was going to say, is that a question?
I think it's a transcript. Yes. Oh, my goodness. That's rather swish. Thank you very much, Paul, for setting the scene. We're moving on to something slightly different now. So obviously, when you're thinking about GBS and you're thinking about vaccines, you have to know the types of GBS that might be important. So Kay's going to be talking to you about that. Over to you.
Thank you, Kirsty.
So I'm going to be talking about the Gbs epidemiology that's found around the world, and also genomics in terms of strains found in GBS. So as Paul has described, GBS is a huge burden around the world. It's found in many, many different regions, but an estimated 21.7 million pregnant women around the world are colonized with GBS. Maternal colonization is the biggest factor for infant to have disease. And you can see in this diagram here from a systematic review that Kirsty, Paul and Neil were part of, you have different prevalences around the world. So the burden of GBS is much higher in low and middle income countries. So in countries such as South Africa, you have a 30% colonization in pregnant women. In the UK, it's about 20%, and lower in Asian countries. So looking at GBS strains, so as Paul has described, there are 10 different serotypes of GBS, but 98% of global strains are due to serotypes 1A, 1B, 2, 3, and 5. But there are regional differences, as I showed you in the previous slide with serotype, with the prevalence of GBS. So in Central America, the predominant strains are made up of serotype 1 and 1B, 1A and 1B, whereas if you compare that to Western Africa, serotype 5 is the most common. Although serotyping has been incredibly useful, it doesn't really tell you about the genomics. So since 2003, there was a Mlst typing system. Mlst stands for multi-locus sequence typing, that was created and implemented. And this looks at 7 housekeeping genes that is present in all GBS strains. So this is amplification of each of the 7 genes, and then sequence analysis and variation of each of these genes. That allele is given a number assignment. And the combination of these numerical assignments give rise to a sequence type. And so by grouping genetically similar sequence types, that gives rise to a clonal complex. So clonal complex is an umbrella of genetically similar strains. So for example, CC23 is made up of ST23, 24 and 22. And due to the widespread use of the Mlst system, we now know that there are certain strains and clonal complexes that are associated with human disease. So for example, CC17, this is very well known to be dominant in causing early onset and late onset disease. Whereas strains that belong to CC1, 19 and 23, these are associated with colonization. Whole genome sequencing has been used a lot recently in the past decade in GBS, and it's been incredibly useful tracking outbreaks and inferring direction of transmission. It's also been used to define pathogenicity, understanding which genes give a pathogenic potential, and also whole genome sequencing has been useful in identifying vaccine targets. So through the use of whole genome sequencing, there's been a paper that's shown that GBS can be present in the amniotic fluid, and also the detection of the HVA gene, which is dominant in CC17 strains and gives the strain more pathogenic potential to cause sepsis, meningitis, and pneumonia. Whole genome sequencing in GBS has been useful also in identifying and tracking outbreaks. So in a study in 2016 in Singapore, there was an outbreak of consumption of raw fish leading to around 200 people becoming ill. And by the use of whole genome sequencing and understanding the strain, it's been pinpointed that it was a particular pathogenic ST283 strain that caused this outbreak. And this was found in particular species of fish. So consuming sushi is still very safe. Also in a hospital setting, in a study that was led by Alita, there was a cluster of a few babies from a hospital that developed late onset disease, and through genomics, it was found that it was because of a particular strain that these babies had in common. GBS was and is predominantly it has origins in the bovine family. So it was originally a cow pathogen, and through sequencing of many strains in the human population and in the bovine population, it's now understood that the strains in the human population were due to strains that harbored a tetracycline resistance gene. And so that population of strains have expanded in the human population. And so that's why over 80% of strains that you find worldwide in humans do have tetracycline resistance. And also, it's not just cows and humans that GBS is found in. It's found in a lot of other animals as well, elephants, dogs, crocodiles, animals that you didn't really think GBS would be colonizing in. So during my PhD, I investigated GBS strains found in humans in the Gambia, and to understand a bit more about the lineages and strains that are found there, and using a cohort of mother-infant pairs, isolating GBS from several different anatomical sites, we found that there are a lot of diverse strains found in the Gambia. So all of the strains were either serotypes 1A, 1B, 2, 3, 4, or 5, but also you had quite a lot of genetically diverse strains. So the strains were associated with human asymptomatic colonization as well, and also there was presence of strains that belonged to the known as the hypervirulent clonal complex. And putting this research into a vaccine context, so as Paul had described, there is a hexavalent vaccine in clinical trials at the moment. And so if that was to be implemented and if it was successful, then one of the it would give 100% coverage of the strains that we found in the Gambia, as all of the serotypes that we found are covered by the hexavalent vaccine. The other vaccine that is a strong contender is the Gbs Alp vaccine. So that's the protein-based vaccine. So in our context, we found that if that was implemented, it would provide an 80% coverage of the strains that we found in the Gambia. So at least 80%, at least one of the rib proteins were found in 80% of the strains that we found in the Gambia. Whole genome sequencing is exploding at the moment in terms of applying that to whole in GBS. So there is a global effort. It's called the Juno Project, that's led by the Wellcome Trust Institute, which Kirsty and St George's is collaborator of. And this is sequencing of as many strains as possible around the world to understand what the genetic landscape of GBS is. We've just sent over 1,000 isolates contributing to this project, and so in the near future we'll be getting lots more information about the genetic landscape of GBS as well. So watch this space. Thank you very much.
Thank you, Kate. Hang on, because you might have questions. Any questions for Kate? Questions online? I can't see any hands up.
Why is it less in Asia?
We don't, so it could be due to several factors. So surveillance may be a crucial factor. So we tend to find there are more surveillance projects happening in USA, in Europe, but there are more surveillance projects happening at the moment. So we may get more up-to-date information about colonization rates. That's all I can think of at the moment.
It wasn't a complete lack of data, right? This data that is.
Yes, yeah.
Any other questions for Kate? Great, thank you very much, Kate. So moving on from whole genome sequences to identification of GBS through molecular methods, is that right, Jason?
That's reasonable enough, yeah, thank you.
Okay. Over to you.
Great, thanks Kirsty. So yeah, I've been asked to come and talk about the work we've more recently got into using the genomics that Kay's described to kind of inform a more sort of diagnostic investigative approach using microarray. So it's really genomics informed sort of approach. And I'm obviously from the Institute of Infection Immunity here and also representing Bugs Bioscience, which is a not-for-profit company of St George's that supports a number of these different projects. So Kay and Paul gave a very nice introduction from using the same information from the CID and looking at the importance of maternal colonization. So really focusing on maternal colonization and infant colonization. And maternal colonization is really the staging post for everything else that happens around GBS. Either the mother may remain healthy, or she may become ill, have impairment, or herself may die. It may then go on and transfer to the fetus, may cause sort of stillbirths, may then be passed on to the neonate through vertical transmission during delivery, cause all sorts of different impairments, premature death, neonatal death that we've heard about already, so the global burden of disease. But that GBS colonization, the mother is really the staging post for all these kind of onward things. And so the more we can understand about that maternal GBS colonization, if we could eradicate GBS maternal colonization, then no more disease will occur. And that's kind of you can't have disease without first having that carriage. As we know, as Kay showed quite nicely, the same graph plot here of maternal colonization across the world. So that kind of average around 18% colonization, but the range in different geographic locations. And Kay also showed that when we look at maternal GBS colonizing serotypes, again, there's some variation by region. But when we look at these overall, the current vaccines in the pipeline will cover 95% of these different serotypes. So some slight differences in Asia, which I'll draw you back to later. So that's the kind of a landscape that we're looking at. And as a GBS newbie, a relative GBS newbie, this is all fantastic information for me getting into the field. So I got into the field really on the back of work we've been doing more in the pneumococcal vaccine area. And so we developed a parallel kind of molecular serum microarray method for the pneumococcus, and this has been used in a wide range of different vaccine studies around the world. And probably the pneumococcus is probably 20 to 25 years ahead in terms of the conjugate vaccine they're using as to where we are with GBS at the moment. So we developed this method here at St George's. It went through this Gates-funded evaluation and was shown to be the leading method. And so we've taken a very parallel approach for GBS. And there's a number of parallels between the pathogens and the approaches that we're using. So for the pneumococcus, we've sort of highlighted carriage as a precursor disease. It involves a complex mucosal colonization in the nasopharynx for pneumococcus. Pneumococcus is more complicated in terms of serotypes. There's 100 serotypes, and polysaccharide conjugate vaccines have been produced that at the moment protect against 13 of those 100 serotypes. So in the UK, PCV7, covering 7 of the serotypes, came in 2006, and PCV13 covering 13 of those 100 serotypes in 2010. And this is now introduced through the childhood vaccination programme in the UK, originally using two childhood priming doses and a booster, and more recently shift to one priming dose and one booster. So slightly different to what we do in GBS. And because it's been around for so long, then we've seen a very good impact on vaccine type disease and carriage. We've established very good immune correlates of protection in the pneumococcal world, which is what Kirsty's working toward for GBS. We've seen a great reduction of disease caused by vaccine serotypes, but in serotype replacement and amount of disease caused by those non-vaccine serotypes not covered by the vaccine. And because we have this impact on carriage as well as disease, then we see this great benefit of herd immunity in unvaccinated individuals. We contrast that with Group B Strep, well, a lot of similarities here. So again, carriage is a precursor to disease, has this complex mucosal colonization, but now it's more rectovaginal and then the GI tract as well as nasopharyngeal. There's only 10 serotypes, again, working towards polysaccharide conjugate vaccines as well as protein vaccines, and that's the same with the pneumococcus. The current GBS 6 vaccine covering 6 of the 10 serotypes in development, sort of phase 1-2 trials at this stage, and this would, instead of being a childhood vaccination, be more of a maternal vaccination program. And again, lots of questions here. So will it have an impact on vaccine type disease? We certainly hope so. That's what we're working towards. Would it have an impact on carriage, or maybe less likely because of the maternal immunisation approach? We're establishing those immune correlates of protection. We're looking at the impact of early onset or late onset disease in the newborns. And again, will there be any impact on maternal colonisation, which give this added benefit of any sort of vaccine program? So as Kay's shown, genomics and biochemistry has taken us through the sort of genetic basis of the different serotypes, of those 10 different serotypes of GBS. And here, when we look at a sort of representation of the genes that are responsible for encoding those different polysaccharide structures, shown here on the left, we can see if you just glance at that, there's not really one gene that defines the serotype. It's kind of the combination of different genes. And so the approach we take is to use this genomic information. Using the microarray, we detect which genes are present in the sample, and then the combination of those capsule genes then enables us to determine which of the serotypes is present. Also, it enables us to determine if multiple serotypes are present, and then at what abundance of different serotypes might be in the sample. So the array we've developed is really initially around looking at different serotypes based on those combination of the capsule biosynthesis genes. We're able to detect multiple serotypes, determine their relative abundance, and also detect sort of the variants of serotypes, maybe non-typables and other odd combinations that we haven't seen before, which could be new serotypes. We include on the array, so while we've got the DNA on the array, we can probe for lots of other information. We can look at factors associated with antimicrobial resistance, so looking for different genes or mutations associated with resistance, covering a range of different antimicrobials and antibiotics we might use. We look for some presence of those genes encoding some of the protein antigens that are being used in the protein vaccines, so alpha-like proteins, serine-rich proteins, and we look for other virulence determinants such as pilosilins or the hypervirulent Gbs adhesion proteins. Because again we're looking at the sort of complex sample as a whole, then we have features on there to identify different other species. So a panel of species-specific gene for other key respiratory or gastrointestinal bacteria. So we might be able to detect which species of Enterococcus is in the sample, E. coli, other species of Streptococcus that might be co-colonizing and may be important. And finally, we can add on, based on all the genomics that Kay talked about, we can add on a pangenome component, looking at the rest of the genome of GBS beyond just the capsule locus. And again, this allows us to determine the genetic relatedness of sample and associate and determine sort of MLST lineages, clonal complexes. So we get the genotype as well as the serotype, which can be quite powerful. So if we think of the carriage studies we're interested in, we start with our swab, rectovaginal swab in this case, or vaginal swab in STGG. This is a very complex sample, has a number of host cells, viruses, multiple strains, serotypes of GBS or other species, and other commensals and co-colonizers in that sample. If we start the microbiology process with a primary plate, this may be selective or non-selective, then again we start to reduce that complexity, but even now we might be looking at multiple strains and multiple serotypes of GBS and potentially other co-colonizers depending on the selection we put in place. And if we go all the way through for a good microbiology practice and purify out that one single colony, then here, of course, if we pick one of those colonies, we'd only ever see one single strain or one serotype. And so a lot of the current methods have been using sort of one colony to kind of represent the whole complexity we might expect to see in that sample. And so the approach we're taking is to use that area there in the middle where we potentially have multiple strains, multiple serotypes. We take all of those colonies from the plate, we extract the DNA, put it on the array, and then start to dissect and work out what's present in those samples. So some of the questions: why are we interested in colonization and asymptomatic carriage? It's not disease. So obviously GBS has primarily evolved as a commensal. It's in competition with other co-colonizers, so there's lots of biology in that niche. It's a precursor to disease, an onwards vertical or horizontal transmission. There's potential for vaccine impact on carriage as well as disease, and this is one we may see indirect effects of vaccine. And the prevalence of carriage versus incidence disease for vaccine impact studies is much greater. So if you look at a cohort of pregnant women, then 20% of those may be carrying, but of course the amount of disease within that cohort is going to be much less, and so you can have smaller studies looking at the impact of carriage compared to the incidence disease. But again, that's only if the vaccine is going to have a reliable impact on carriage. But in the pneumococcal world, carriage has been used as a proxy now to look at vaccine impact. Why multiple serotype carriage? So current methods may be missing carriage of additional serotypes at low abundance. This can impact on models of acquisition, duration of carriage, transmission and competition between the different serotypes. We've seen an indication of multiple carriage from many different methods, maybe taking multiple colony picks, sequencing multiple colonies or serotyping them, and also based on morphology differences. And sometimes multiple carriage can explain discrepancies we see in between mother-infant pairs. So maybe the mother has a different serotype to the infant, then you think, well, how did the infant ever get that? But of course, if the mother's carrying multiple ones and only one of those has been passed on, that explains those sort of discrepancies. Likewise, if we do repeat serotyping of samples, if we pick different colonies from the mix, just by chance, we may pick different serotypes at different times. So we're trying to capture all that complexity. So I'm going to skip through really the initial proof of concept work. We looked at the PHA reference strains for the 10 serotypes. They all worked well and confirmed what we expect to see. We did some isolates from a carriage study in the Gambia with Kirsty. We saw some discordance here between the microarray result and latex results, although generally the microarray results were concordant for the isolate and a matched swab. And again, when we looked and compared to PCR results, microarray tended to agree more with PCR than with latex. And I'll show you some data while we have confidence in our result. Obviously, as a new method, people always imagine that, well, you don't know what you're doing and you're getting it wrong. But hopefully I can prove that we do know what we're doing. And we looked at then at some swabs collected from the carriage study in the Gambia. So again, 19 of 51 swabs, around half of the swabs had multiple serotypes in them. 10 had 2 serotypes, 5 had 3, 4 had 4 serotypes. So plenty of multiple serotype carriage in these swabs. This meant we had detected an additional 32 serotypes in these samples by microarray. 51 was found by latex, but 83 were found by array. And again, we were able to detect other species in these samples. Just to look at some of those discordant results, this is an example where the latex serotype was reported as serotype 8, but by microarray it was serotype 2. And here, just looking at the raw data, the dots in red are what we expect to find for serotype 2. Those in yellow are what we'd expect to find in serotype 8, and they're sitting down in background as negative. So all the genes we expect to find for serotype 2 are present. And likewise, another example here where this is actually showing multiple carriage of the serotype 5 and serotype 1A by microarray. We can see them there at different abundances. The 1As there are about 7%, whereas latex had reported this to be a serotype 2. But again, all the genes we'd expect to find in serotype 2 are down in background and not present. So we have reasonable confidence in our calls, and there's lots of reasons why we may see these discrepancies. Again, using the microarray data, the genome component, we can generate these trees. And again, we can use to look at this discordant samples. So here they're clustered by the genotype, the genetic backbone, and they're coloured by serotype. So we can go through, and using this, we can look at those matched samples where there's discordance, and generally where they match up, they match other samples of the same lineage or from the same swab that are of the same serotype. So there's lots of evidence we're getting pointing to the fact that actually our results seem to be pretty reliable. So we've gone on to use this now in a number of studies. So in the Kirsty samples in the Gambia, we've looked at mother rectovaginal swabs and infant rectovaginal and nasopharyngeal swabs taken at delivery or birth. In the mother swabs, there's sort of 31% of samples have multiple serotypes, whereas in the infants it's slightly higher at 52% of samples, although we have fewer samples. And overall here you can see actually the serotype distribution is very similar between the mother and the infants, pretty much identical as we look at it by eye. However, if we drill down into this, if we looked at those we detected as the major serotypes, so these would be the ones we detected by chance if we just picked a colony, then we could see quite a different profile, quite a different sort of distribution of serotypes between the mother and the infants. But then when we look at the minor serotypes we're detecting, those we're detecting in addition to the serotypes, then we can see where those other serotypes are. So for instance, some serotype 3s and 1As we're only detecting as minor serotypes in the infants, so they're still present. So this has given a much more complete picture of what's present in these different populations and the advantage of detecting minor as well as major serotypes. Another study we've undertaken is now multi-centre studies, so looking at samples for a range of different sites with a group in South Africa. And again here, a lower range generally of multiple serotype carriage, so only around 8 to 12%. However, these are vaginal swabs and not rectovaginal swabs as we saw previously, so that may explain some of these differences. The good thing with this, when we scan through it, is actually the vaccine, so the 6 serotype based vaccine, the majority of these sites would cover 100% of the serotypes we're seeing. Only in sort of Kenya and Nigeria, it drops slightly to sort of 99% of strains, and slightly lower in Bangladesh, which is the only Asian site we've looked at to date, but still around 95% of the serotypes we're seeing. Again, looking at the serotype distribution from those different sites, these are similar to the plots we saw earlier, but now with the data we've generated, and we can see we're going to get very good coverage with the 6-valent GBS polysaccharide vaccine. And just really those in purple you can see there in cyan, just those few serotypes that wouldn't be covered by the current vaccine. So I hope I've shown we've got proof of concept for this as an approach, very robust genomics-based methodology, and the benefits of multiple serotype detection. We've evaluated a number of multi-centre studies now and compared it to other methods being used, and we're looking to use further work to calculate colonization density and look at some of the other features that we've mentioned. So thank you for your interest. And there's the acknowledgements.
So when you implement the 6-valent vaccine, how long do you think it will be before the other 4 serotypes will emerge as important strains?
With GBS, we don't know. In the UK and Pneumo, it's probably been 5 or 10 years. But in GBS, we're not seeing the other serotypes in some types. So where would they come from if they're going to be replacement? And also, we don't know if there's going to be any impact on carriage. So if there's no impact on carriage, we probably wouldn't see those serotype replacement effects in the same way. But yeah, where would they come from? We're not seeing them in some of these sites, but it's something that would be monitored for sure. But certainly in the UK, it's been quite a dramatic serotype replacement effect.
Tom, just a rather practical question. Who are your partners in the NIH Mucosal Pathogens Research Unit? Is that where the money is coming from?
Yeah, so that's based at UCL with Rob Heidemann and David Goldblatt.
When was that? When did that start?
It's finished.
5 or 6 years ago, actually.
Groups.
Right.
Yeah.
So are you going to reapply? Is that
They
important for you to carry on?
We've missed this round. We didn't get one. We did apply, we didn't get it. But yes, that would have been a good idea.
But there's other funding streams now coming through around the vaccine anyway. So we've used that money really to do the proof of concept and show some of the initial studies. So it's not a showstopper.
No, exactly.
Yeah.
Does it help you or hinder you, the fact that you're not for profit bugs group? Does that help funding wise?
I think it helps when we work with partners like Gates and Path And it helps a partner like Gates and Path and people in Gavi, then it just sits better with them, I think, really. So, yeah.
There we go, mood lighting and a temperature warning. Thanks, Jason. There's no questions online, were there? Yes, there is. There's a hand up.
It's me.
Hello, Jodie. Hi.
Jason, that was super clear. That was really helpful. I was curious about how much multi-serotypes you have in the samples, and is the capsule region stable, or is it really tightly aligned with the sequence types? So how much variation are you going to get, and any idea what that might do in terms of escaping vaccines?
So again, we don't know in GBS, but from Pneumo there have been examples of serotype switching, where serotypes can switch, so you keep the same genetic background, but the capsule loci will recombine in and change. That could be under vaccine pressure, or it could have just been there all the time and vaccines selected out those different variants. So it's a possibility, but we don't know what would happen with GBS at this stage.
Okay,
and I didn't get the first part of your question, Jodie, I don't think.
Yeah, I was wondering how closely linked the serotypes are with the sequence types.
Yeah, so yeah, reasonably close link, but it's not exclusive. So different serotypes will have different genetic backgrounds, different lineages will contain different serotypes. So they're reasonably constant, but they can switch about and they can change. And with recombination, then you can have serotype switching. So
great, thank you.
Thanks.
Thanks, Jodie. There's no other hands up. Thanks, Jason. So moving back now to our final talk, back into the clinical side of GBS with Costas, who is at home today, so he's presenting online. Costas, have you got slides to share?
Yes, let me share my screen.
There we go. Did that work?
Yep.
And Jodie, your hand's still up unless you had a second question for Jason. Thanks.
Can you see my slides okay?
Yes, perfect.
Great.
How do you make them full screen? Is that do you have to make them full screen? Is there something I have to push? Full screen here, yeah. Can everybody see it? Over to you, Costas.
Oh, good. Thank you, Kirsty. So, hello, my name is Costas, and I'm a clinical research fellow at St. George's. I'm really grateful for the opportunity to talk in the Jennerosity event today. For the next 10 minutes, I'm going to go through some recent research on the understanding of risk factors and pathogenesis of late onset GBS disease. So historically, invasive GBS disease in infants is divided into early onset from birth through day 6 of life, and late onset from day 7 to day 89 of age. And there is a good reason to think that these two clinical entities are distinct. Early onset results from vertical transmission at delivery or shortly before from a mother colonized with GBS. Risk factors for the early onset disease have been well described and include maternal colonization, prematurity, prolonged rupture of membranes, intrapartum fever, among others. On the other hand, the pathogenesis of late onset disease is not very well described, is less well understood, and the transmission routes less well described. The clinical presentation might also differ, with a higher proportion of late onset cases presenting as meningitis. The most worrisome, though, epidemiological observation is that late onset disease rates have not changed with the widespread use of intrapartum antibiotic prophylaxis. Data from the United States, shown here on the left-hand side, clearly show that intrapartum antibiotic prophylaxis has substantially reduced the incidence of early onset disease, but made no impact on late onset disease. Data from France, shown here on the right-hand side, of approximately from the same period, also show that early onset meningitis cases, here the green dots, have gone down by 63%, whereas late onset meningitis cases, the red dots, have gone up by 58%. And similar findings have also been reported by other European countries like the Netherlands and Iceland. So compared to early onset, the risk factors for late onset disease are less well studied. So to address this gap, we did a systematic review. Paul and Kirsty have been involved, and our meta-analysis showed that preterm infants have almost 6 times higher risk than term infants in developing late onset disease. And this risk is increasing for each week of decreasing gestation. So why are preterm babies at higher risk? First of all, the placental transfer of IgG antibody is optimal in the 3rd trimester of pregnancy. Therefore, infants born prematurely might not have had a chance to receive protective concentrations. Also, lower gestational age is associated with less effective phagocytosis and less effective opsonic neutrophil-mediated defenses. In addition, premature and low birth weight infants have increased gut permeability and a higher risk of nosocomial transmission because of prolonged hospitalization. Prematurity is also characterized by disturbances of microbiota as a consequence of frequent antibiotic use, in formula feeding, and restricted contact with the maternal microbiome. And this might disturb the adaptation of GBS to its neonatal host environment, leading to dysbiosis, barrier disruption, and GBS dissemination. The other important finding from our systematic review was that babies born to women colonized with GBS antenatally have almost 3 times higher risk of developing late onset disease compared to babies born to mothers non-colonized with GBS. Now, this finding could be explained by the dynamic nature of GBS colonization. Mother-to-infant transmission of GBS can take place for several weeks after delivery. A study from Italy showed that 20% of babies born to mothers colonized with GBS were also became colonized with the same strain as the mother by 1 month of age, despite adequate intrapartum antibiotic prophylaxis and a negative GBS screening at birth. A study from Japan has also confirmed the same findings. And in addition, a study from France showed that this is also serotype dependent, and the clonal complex 17 is particularly linked to an enhanced post-delivery mother-to-infant transmission. So these observations taken together suggest 2 modes of GBS acquisition in early life. A vertical transmission that is lowered but not fully suppressed by intrapartum antibiotic prophylaxis, and that then later presents as late onset disease, or delayed horizontal transmission after delivery that likely occurs through nursing, for example, with via contaminated hands, or possibly breastfeeding, although the latter remains quite controversial. Another important transmission route that is poorly recognized is the nosocomial transmission. Hospital outbreaks of invasive GBS disease are considered to occur infrequently. However, genome sequencing of 410 isolates in infants from the UK and the Republic of Ireland from the national surveillance study that was led by Paul showed that revealed potential seven potential clusters of infant GBS disease in neonatal intensive care units, 4 of which had not been previously detected. So the authors of this paper drew the conclusion that routine submission of GBS isolates to the reference lab for genome sequencing could actually help in detecting potential clusters. So far we have focused mostly on the host factors. What about the pathogen? So late onset GBS disease is a disease caused mainly by serotype 3 and more precisely by clonal complex 17, a particular hypervirulent GBS clonal complex almost exclusively of serotype 3. And the recent systematic review that Kirsty has been involved of the global serotype distribution showed that although serotype 3 is found in 28% of colonized women, it is responsible for 77% of late onset cases. In other words, it causes more cases of invasive disease than expected from its colonization rate of pregnant women. So why is that? Clonal complex 17, which is also linked to most cases of GBS meningitis, expresses several virulence factors. And the most important of which are the adhesin protein HVGA and SRR2, which have been associated with enhanced adhesion to epithelial cells and host plasma proteins. US data showed here on the left-hand side, on the figure on the left-hand side, showed a rise in the incidence of serotype 3 associated GBS disease since the early 2000s. Similarly, the increasing incidence of GBS infections in the Netherlands over the same period was found to be associated with a clonal expansion of CC17, as it is represented here from the dark green color in the middle figure. And this was correlated with the emergence of 2 new sublineages, A1 and A2, represented by the light blue and the dark blue colors, respectively, in the right figure. Especially A1 was correlated with the acquisition of a new phage, which was first acquired in 1997 and increased steadily from 2000 onwards. So what can we do to prevent this from happening, given the shortcomings of intrapartum antibiotic prophylaxis? As we've heard from Paul, a vaccine administered during pregnancy could substantially reduce the disease burden. Given that 3/4 of all late onset cases occur within the first 8 weeks of life, and that the half-life of vaccine-induced antibodies based on phase 2 trials so far probably would be between 39 and 46 days, most infants would be protected. However, infants born prematurely may not have a chance to receive protective concentrations, and that's a big challenge. Because these are the babies who are on high risk, as we said. However, this will be strongly influenced by the timing of vaccination during pregnancy. And we know from several recent studies on maternal vaccination against pertussis that they support the efficacy and the efficiency of early vaccination during pregnancy. So a study by Kent and colleagues showed that preterm infants born to mothers immunized from 28 weeks had higher antibody concentrations than preterm infants born to unvaccinated women. And another study by Eberhardt and colleagues also reported higher antibody concentrations in preterm babies when mothers were vaccinated in the second trimester compared to the third. And the vaccination effectiveness data from the United Kingdom suggested that expanding the window down to 16 weeks of gestational age in the UK reduced the hospitalized pertussis case in preterm infants. Therefore, in the future, after we have a vaccine, studies trying to define the optimal window for GBS vaccination should be prioritized. To sum up, there are still significant gaps exist in the knowledge of late onset GBS disease and the knowledge of pathogenesis of late onset disease. Prematurity and maternal colonization are major risk factors for late onset disease. Maternal vaccination is likely to be the best preventive strategy for the future, but in the meantime, enhanced surveillance with genomics to monitor disease trends and characteristics is really of paramount importance, as it is shown with the expansion of CC17 seen in other countries and the increased recognition of hospital clusters. I'd like to thank my supervisors, Paul and Kirsty, and all the lovely people from our GBS lab. Thank you very much. Happy to take any questions.
Thanks, Costas. Are there any questions either online or in the room for Costas? Jane has a question for you, Costas.
Hi, Costas. That was fascinating. Thank you. And I guess one question, I was wondering if, obviously, about late onset and whether babies pick up the bacterium from to develop late onset Group B strep infection. I was wondering if there'd been any research into the role of fathers. We hear a lot about Group B strep being passed on from the mothers and the mothers' colonization status. And I just wondered if there's anything. I haven't seen anything, so you may well, but I don't.
I haven't seen anything, although I think there might be something from very old studies, and I think Kirsty knows better the answer to this, but this is definitely a very significant gap in the literature and something that we should probably try to do better. So when we do this kind of studies with the carried studies, we should probably look on not just the mother, but also the father and the extended family. This is, I think, this is a significant gap in the literature and very important question.
Yeah, I agree. I think we also have to be very careful that we can associate causation, because otherwise I think it creates a lot of anxiety, as you well know. But yes. Any other questions for Costas? Lovely. Thanks very much, Costas. Thank you to everybody for attending. And Julian, do you want to close the session? Or?
Well, no, I'll let you.
Oh, okay, fine. Thank you to everybody for attending, and thank you again to everybody here and online and at St George's for buying raffle tickets, making cakes, talking to people about GBS and raising the profile of Group B Streptococcus. I hope today, if you didn't know what GBS was, that you're a little bit more convinced of how vitally important research in this area is. And thank you very much to GBS for allowing us to support you. Thank you. So those online, will leave your cake upstairs for when you're next in. And everybody else, we'd like to invite you up to the INI for cake and the grand raffle draw. I know you can't wait.